Gene editing has been developed over many decades with limited success, that is, until the advent of CRISPR-Cas9. This gene editing technology hijacks the mechanisms of bacterial immune systems, enabling us to delete undesirable genes and insert desirable ones with extreme precision. CRISPR was catapulted into the history books in 2018 when Chinese researcher He Jiankui controversially created the world's first genetically modified babies with little regard for bioethics or regulation. This outraged the international community, sparking legislative and moral debates. Despite this shaky introduction, CRISPR presents a promising future for medical research and therapies. However, it must be monitored on a national and international scale to ensure legal and ethical use.
Gene editing has had a bumpy history since the 1970s when scientists bombarded DNA with assortments of genetic tools, most of which were not studied thoroughly. Since then, CRISPR has provided us with the most reliable and efficient methods of gene editing yet. Conventional gene therapy is designed to introduce genetic material into cells to compensate for malfunctioning genes or to make a beneficial protein. While gene therapy has been tested since 1989, the process of randomly inserting genes is highly unreliable and may have taken years to complete. As such, the search for a fast and more accurate version of gene editing started, and the crude idea of CRISPR was proposed. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, was first coined by Francisco Mojica in the early 2000s, where he recognized that the genomes of bacteria and archaea featured the genomes of bacteriophages, and therefore the CRISPR DNA sequence acts as an adaptive immune system. The idea of modern-day CRISPR-Cas9 has largely been credited to Jennifer Doudna and Emmanuel Charpentier in 2011. They discovered how the adaptive immune system of CRISPR functions. Ultimately, Emmanuel's research gave us the final piece of the puzzle in order to utilize CRISPR for efficient gene editing. In 2018, researcher He Jiankui caused major footing in the world of CRISPR when he controversially made genetic modifications to newborn twins. His experimentation can be considered an unconfirmed scientific breakthrough and opened the gates to public domain. This larger audience engaging in the Jiankui Affair has caused further scrutiny and attention within the field of genetic engineering.
We've now reached a point where the CRISPR system is so advanced we can't even begin to tap into its full power. CRISPR takes advantage of a process within bacterial organisms, which lack a complex immune system, so instead rely on repeating sections of DNA called CRISPR arrays. These segments of DNA capture, identify, and remember the viruses that have compromised the bacterium in the past. Using this DNA memory, the bacteria is able to identify the same virus, create a specialized section of guide RNA which targets viral DNA, then attach an enzyme called Cas9 to this guide, which cuts the virus of the DNA to destroy it. By creating a length of synthetic guide RNA, geneticists can attach their customized segment of information to the Cas9 enzyme, which then cuts the DNA exactly where they want it to. Following this, researchers are able to hijack the DNA repair mechanism of the cell to replace the removed gene with a specifically created one, effectively replacing undesirable DNA with synthesized DNA. Now, there are two approaches to human genome editing. The first is gene therapy, which corrects genetic errors in a person with diseases such as cystic fibrosis by gradually changing the genotype of their body's cells. Few would argue against this technique, as the therapeutic benefits are unparalleled for these otherwise incurable conditions, and the genetic editing remains confined to just the original, consenting patient. The other, much more controversial type of genome editing is germline gene editing, which is when researchers edit sperm or ova cells to change genes passed on to a child, as well as any descendant of that individual. You might have heard of this process in the media, when designer babies are discussed. This term is used because researchers could potentially genetically engineer individuals who are predisposed towards athleticism, intelligence, and overall health, as well as possessing certain cosmetic traits. While this is technically possible, the human genome is incredibly complex, and the way our DNA affects many traits is not yet understood. If an error is made in the editing process, the potentially tragic outcomes of this failure are irreversible, as the error would be in every cell of the child.
This exact concern was realised in 2018 with He Jiankui's CRISPR twins, the first children in history to be born with CRISPR-edited genes. He edited the CCR5 gene to give them immunity to HIV/AIDS, ignoring all government bans. This act was the catalyst for CRISPR's entry into the public domain. By inciting worldwide outrage, he cemented the scandal in biomedical history. The girls were born without the disease. However, some genetic issues occurred. For example, one of the twins has some cells with altered genes and some without. The long-term impact of these modifications will not be known for years to come, and it is this uncertainty that is most concerning. The case also represents the crossover between enhancement and therapy, because as well as HIV immunity, the CCR5 gene has been linked to intelligence in mice. As the line between enhancement and therapeutic modification becomes increasingly blurred, the question is not whether we can do this, but whether we should. If the CCR5 CRISPR editing process worked perfectly, would we have the medical responsibility to remove HIV from the human race, just like we functionally eradicated polio with the global vaccine? This concept is highly controversial for conditions like autism and dwarfism, and the right of medical professionals to decide on the worthiness of a human life. This mirrors the practice of eugenics, the removal of undesirable qualities or conditions from the human genome, which has occurred throughout history, for example, in the generational whitewashing of Indigenous Australians. We now hold a powerful, cheap, and accessible biotechnology in our hands. Cases like Jiankui's remind us of just how important it is to monitor its use. The ethics of editing traits, disability, disease, and immunity, both in and out of genomes, will remain a major limitation on CRISPR technology long after it is determined safe to use by clinical trials. The world of biotechnology is entering morally uncharted waters, and very little ethical precedent or legislation exists to control it.
As ethical issues and reliability of CRISPR are contentious, legislation should be well governed and defined to prevent complications. However, in 2016, when Dr. He Jiankui did decide to open the door to germline editing, no official regulations were in place. Ultimately, Jiankui's advancements were received with public outcry due to ethical concerns, illustrated in the statements of the Science and Nature journals that modifying embryos should be off limits. Accordingly, this controversy led to an international discussion on current gene editing laws and regulations. Legal repercussions were seen immediately after the incident, when Chinese authorities announced that he was found guilty of forging documents and of unethical conducts, sentencing him to 3 years in prison with a fine of 3 million yuan. Additionally, the Zhejiang University swiftly made a press release outlining the failures of the ethics governance system at the time, which largely relied on self-regulation. As a consequence, the World Health Organization launched a global registry to track human genome editing and put together an expert panel outlining its scientific, ethical, social, and legal guidelines. It was determined from this that editing embryos remains illegal due to such ethical issues that were brought up by Jiankui's failure. As a result, this has made researching CRISPR harder and more costly. Moving forward, regulatory institutions are keeping a close eye on what CRISPR can do. Until then, we must rely on newly established rules that arose due to incorrect and immoral advances with this technology. There is no doubt that genetic engineering will save many lives and render many current diseases curable. It is also conceivable that public access to CRISPR may occur during our lifetime. However, until CRISPR's accuracy is improved, it has been determined by the World Health Organization that genetic engineering is not permitted.
As mentioned before, CRISPR isn't perfectly accurate. The risk of off-target cuts to the DNA sequence and subsequent mutations are a major point of contention, and a reason the HIV baby situation wasn't taken well by the scientific community. Current off-target prediction methods require computational software and are relatively unreliable, so future research is looking into how artificial intelligence could be employed to identify and reduce off-target effects. By feeding genomes into these AI systems and initially helping them to identify sites where off-target cuts may be made, machine learning can then be used to continuously improve the ability to detect off-target regions, hence minimizing the potential splicing in these areas. Public opinion is generally influenced by the intentions behind these experiments. Australians tend to be morally against tests involving animal gene editing to improve human food sources. He Jiankui had explicitly stated that CRISPR in humans should be used to eliminate genetic diseases. Concerns for the accessibility and transparency of public versus privately funded research plays an enormous factor in the future of CRISPR, especially since Jiankui's actions have created a negative stigma around CRISPR and Chinese research focusing it. In future, it would be more socially acceptable for the research to be conducted through public funding or by universities, rather than private corporations, even if the same interests are to be pursued.
Historical milestones made by Charpentier, Doudna, and Jiankui have been monumental in setting, as well as paving, the ethical and legal future of CRISPR. The science of the bacterial immune systems have been adapted to human use after decades of development. The controversies surrounding Jiankui's research have not only spread awareness about the gene editing tool, but also given a glimpse at public reaction towards the future of cutting-edge medical advancements. Currently, the moral and ethical standards are the limiting factor in the acceleration of CRISPR research, but perhaps with international regulations and ethics committees agreeing upon this set standard of practice, the rate of new discoveries could be unlike anything we have ever seen before.